Dr. Mamta, you can start introducing her and then she can start.
Thank you, sir. Good morning to all of you. I personally thanks the organizer for giving me this opportunity to introduce Dr. Shweta, who is very dear to me. So, Dr. Shweta, currently working as assistant professor in College of Veterinary Science, Dovasu, Mathura. Dr. Shweta, born in Kanpur city in Uttar Pradesh in year 1986, after completion of her schooling in the same city, she graduated BVSc and AH in year 2016 from Duwasu, Mathuras with a first division. Dr. Sachan later qualified ICAR JRF for master's degree program and completed her master's of veterinary science in the discipline of animal genetics and breeding from NDRI Karnal, Haryana in year 2018, again with first division. Dr. Shweta Sachan secured fourth place in Indian Veterinary Research Institute, Uttar Pradesh entrance examination for her PhD degree program. She has awarded institutional fellowship and did her PhD degree in AGB in year 2021. Dr. Sachan has qualified ICAR National Eligibility Test and SRF in year 2018. Her research work on genetic analysis of lactation persistency in dairy animal received considerable acclaim. She has published 6 research articles, 2 review articles, 2 books, 11 laboratory manuals, and 6 popular magazine articles. She is a lifetime member of Animal Nutrition Association, Indian Poultry Science Association, and Society of Conservation of Domestic Animal and Biodiversity. She has won various quizzes and awards in various competitions. Dr. Sachan is a recipient of Best Young Scientist Award by Society of World Environment, Food and Technology, PhD Best Thesis Award by Indian Society of Agriculture, Science and Technology for Rural Development, Young Scientist Award by Society for World Environment, Food and Technology, and so on. So for today's her topic of delivering is significance of genetic approaches in selecting high producing animal, and I'm sure the participants will highly benefited with her presentation. So over to you, Dr. Shweta.
Thank you, ma'am. Thank you so much, ma'am. A very good morning to one and all. Myself, Dr. Shweta Sachan, and welcome to my presentation. First of all, let me thank you all for joining here today.
Ma'am, you stopped presenting. Hello?
Yes, sir.
You stopped presenting. Can you start
No,
presenting again?
sir. There is some net problem. Just I am starting, sir.
Okay. Can you share your screen again?
Yes, sir. Yes, sir.
Start the slideshow again. Your screen is visible. Can you click on the slideshow?
Just a minute, sir. Wait.
Sure. You're sharing a window actually. That is the problem. You have to share the entire screen. Share the entire screen. That way your slideshow would also be visible. Choose the third option, entire screen, rather than the second one. Can you unmute yourself now? Yes, now you can continue. The slide is visible. You can continue.
As you can see on the screen, my presentation topic today is significance of genetic approaches in selecting high producing animals. In my presentation, I have focused on major four sections. First, selection, then second, type of selection, third, selection procedures, that is further differentiated in three categories. First, traditional selection, then marker-assisted selection, and third one is genomic selection, and last, conclusions. Now first, selection. What is selection? As we know, selection is a differential propagation of genetic material to the next generations. And there is a changes the gene frequency and genotype frequency without creating any new genes. And the selection is generally depends upon the sampling nature of inheritance and having process, because in every generation genes become same but their genotypic frequency halves, and it is predicted in the form of response to selection or genetic gain. That is predicted in a formula. Response to selection is equals to heritability and selection differential. When heritability is high, then response to selection or genetic gain is high. If heritability is zero, then response to selection is zero. As we know, selection is of two types. First, natural selection, and second, artificial selection. Natural selection is basically a survival of the fittest. In picture, we can see that sheep are easily adapted for high climatic region or temperature. But in artificial selection, it is a man-made selection because there is a human interventions in breeding animals for a particular trait or specific trait. And in picture, merino is generally developed by artificial selection. Now third, selection procedure. As we know, there are basically three methods. First is traditional selection, and second, marker-assisted selection, and most advanced or genetic selection, that is a genomic selection. First, traditional selection. Traditional selection is basically depends on the phenotypic record, that is a pedigree record and a progeny record, and these records is used for the estimation of expected breeding value of the animal. There are different types of traditional selection. First, individual selection, family selection, within family selection, sheep selection, and finally progeny testing. But there are still some limitations. First, low heritable traits and sex-limited traits are not generally selected by the traditional method. And there is a continuous record of phenotypes. That is a very hectic process of that. Or the records of animals is some difficult to maintain. And last, young animals, because young animals are not directly selected by the traditional selection because there is no availability of the records. And the most important, that long generation interval, because in traditional selection method, generation interval is very large because age at first calving or generation interval in cattle and buffalo is large. Now we move on to the second, marker-assisted selection, that is a type of genetic selection. It is the indirect selection of genetic determinant of trait of interest by using a markers. Different types of markers are used, like molecular markers, biochemical markers, and chromosomal markers. Marker-assisted selection is a breeding procedure in which DNA marker detection and selections are integrated into a traditional breeding program. And there are basic two assumptions during marker-assisted selection. First, DNA markers can reliably predict the phenotype. That is a perfect marker, and they are tightly linked to the loci. And as we know, gene is directly related with certain phenotypes, and markers are genetically linked to a gene of interest. If a gene is not isolated, markers helps to tag a gene of interest. The basic properties of markers should be codominant and polymorphic. It should be demonstrated in all types of tissues, and markers should be abundant in the populations. There are different three methods that is used for the detection of QTL, that is a quantitative trait loci. First, gene-assisted selection. Second, linkage disequilibrium marker-assisted selection. And third is linkage equilibrium marker-assisted selection. In all these methods, gene-assisted selection is best method for the detection of QTL because genes are directed associated with the trait of interest. Now, response to selection. Response to selection by marker-assisted selection. In this graph, we can see that by normal selection, there is a short-term benefits as compared to marker-assisted selection, because in marker-assisted selection, genetic gain is about 10 reached to 2 to 60%. And it is a mainly used for the traits with low heritable traits like reproductive traits, and it can be also used for the with few recordings because there is some expensive recording for the trait, and it can be measured late in life such that traits recordings are not available at the time of selections. Marker assisted selection is also used for the slaughter call to traits like carcass trait, and it's also used for the disease-resistant trait because disease-resistant traits is a low heritable trait, and that can be not directly measured by the traditional method. Now, marker-assisted selection used in selection program will be used between breed selection and within breed selection. Between breed selection, marker-assisted selection is used for introgression program. In introgression, there is a movement of the target gene from the donor breed into the gene pool of the second breed. And the best example of marker-assisted selection in between breed selection, first is neck and neck gene in poultry and Borola gene. And second, marker-assisted selection is used within breed selection, that is the improvement of synthetic lines. Tandem selection with selection on QTL followed by selection on polygenic estimated breeding values. Thus, marker-assisted selection will complement to the conventional breeding program. There are some advantages of marker-assisted selection. First, it increases the selection accuracy and increase in selection response because there is more genetic gain as compared to traditional method. And marker-assisted selection can be used at a very young age of animals. But there are still some limitations because there is a low marker density and the detection of QTL is very difficult. And genotyping marker is also a high cost. That's why now present day genomic selection is used. Genomic selection is a type of genetic improvement. As we know, genetic improvement requires sophisticated approach as a large majority of relevant traits that are quantitative traits and genetically interconnected in both favorable and unfavorable conditions. Therefore, sustainable breeding goals requires the measurement and proper weighting of all relevant traits in selection indexes to enable simultaneous genetic progress in the desired direction for all traits of interest. Because in genomic selection is a form of marker-assisted selection. But in genomic selection, genetic markers covering the whole genome that are used for the all quantitative trait, and these all quantitative trait are in linkage disequilibrium with at least one marker. And by the use of genomic selection, genomic estimated breeding value is calculated from all the dense markers across the whole genome. Now, why genomic selection is very important for animal breeding? Because in genomic selection, genome-wide high-density markers information used through the SNP chips and low cost of genotyping by the DNA sequencing, and there is a high accuracy in estimation of breeding value of individuals. There are some indications where genomic selections are used. First, low heritable trait and sex-limited traits. And it is also used for the traits which are very difficult to measure or very expensive. And these are also used to traits which are difficult to improve directly. And in unfavorable genotype environment correlation, if there is a genotype environment correlation is unfavorable for any trait, then genomic selection is used, and it is also used for the young male animals because there is no need of a progeny records. Now, principle of genomic selection. The basic principle of genomic selection is that information from a large number of makers is used to estimate breeding values, that is a genomic estimated breeding value, without having a precise knowledge of where specific genes are located on the genome or not. Around 10,000 SNPs are used for whole genome with an assumption that single SNP is in close to the gene of interest. SNP chips are used for the genomic selection of bovine. Nowadays, Illumina bovine SNP 50 bead chip that contains around 57,000 SNPs uniformly distributed across the whole genome of a cattle. And it includes genome-wide selection, identification of quantitative trait loci, and also use in the evaluation of genetic merit of animals. There are basically three steps to genomic selection in a breeding program. First, animals are both genotype and phenotype in a reference population, and each SNP is estimated by using a statistical method. And then SNP data and phenotypic data are combined to form a prediction equation. And finally, in subsequent generations, young animals are only genotyped to determine effect at each QTL and to predict genomic estimated breeding value by using the prediction equation. In this diagram, we can see there is a reference population and their genotypes and phenotypes are known, and then make a prediction equation by using these genotypes and phenotype. And then selection candidates will selected on the basis of markers genotype. Then these selected animals are used as a breeder for the next generation by using genomic breeding values. Now, methodology for genomic selection. First, use the markers to reduce the genotype of each animal at each QTL, and then estimate the effect of each QTL genotype on that trait, and then sum all the QTL effects for selection candidates to obtain their genomic estimated breeding values. Now, there are basically three methods. First is single marker genotype, and second is haplotype, and third is identical by descent. In single marker genotype, it is in a linkage disequilibrium with QTL, and then proportion of QTL variance, that is R square, is explained by a single marker only. Its accuracy is high when high genetic gain and high density markers. In haplotype, second is haplotype. In haplotype approach, QTL is in linkage disequilibrium with multiple marker haplotypes and high accuracy as compared to single marker genotype. Now, third approach, that is identical by descent, that is mainly used for the QTL marker association, because there is a probability of two alleles to be remain same for common ancestors. And there is a high accuracy even in a low marker density with low heritable traits. Thus, IBD is best method for the detection of association between the QTLs and markers. Now, estimation of QTL effects. Basically, three methods are used: least square methods, BLUP, that is the best linear unbiased prediction model, and third is Bayesian method. In all these three methods, Bayesian method is best for the estimation of QTL effects in genomic selection. First, we see least square method. Least square method is a general simple linear equations where it make a single segment regression analysis and then select most significant segment. But there are still some limitations. First, selection of most significant chromosome segment means there is only a significant chromosome segment can be selected by a least square method. And the correlation between estimated breeding value and true breeding value is very low or low, and impossible to estimate the effect of all QTLs simultaneously because it is a linear equation and it is used for only one QTL at a time. Next, BLUB. BLUB, that is a best linear unbiased prediction model. It is a best method because QTL effects are normally distributed with constant variance and simultaneously measurable. Where this is a genomic estimated breeding value where n is the number of chromosome segment across the genome and xi is the marker genotype and gi is the BLUB estimate of the ith SNP effect. Its accuracy is high in estimated breeding value, but Bayesian method is mostly used for detection of breeding values for genomic selection because Bayesian method with a prior that assumes many QTL have a small effect and few have a large effect. That is the Bayes method A. And Bayes method B, that is use a simple rule about conditional probabilities. In these two methods, Bayesian method B is used generally because there is a conditional probabilities. Now, accuracy of genomic selection. Level of linkage disequilibrium is more. That's why it is more accurate estimation of breeding value of an individual. And in a reference population, number of effective population size is required very less. And its accuracy is also high because density of markers is more. And it is also used for the low heritable trait. Or the traits which have low heritability like reproductive trait. Accuracy of genomic estimated breeding value for a bull calf is high as accuracy of estimated breeding value after a progeny testing. Accuracies of genomic estimated breeding value for young bulls that is between 0.4 to 0.8 across the range of traits. In this slide, we can see that the genomic estimated breeding value by a Bayes method is more as compared to a least square method. That's why Bayesian method B is mainly used for the calculation of genomic estimated breeding value of an individuals. Now, advantages of genomic selection. By the genomic selection, there is a genetic gain is double as compared to traditional selection, and there is a reduction in cost up to 92% in progeny testing because it is a progeny testing is a around 5 years program, but in genomic selection we can easily select the best animal in early age of life and they can be used for the next generations or in a breeding program. And by genomic selection in breeding is also less because there is no more generations is required, and accuracy of breeding value is around 0.85, and it is also highly accurate for low heritable traits for juvenile animals. In this diagram, we can see that genetic gain with marker information is very high at the age of 11, but at the age of 35, it is less. That's why genetic selection is directly done by the genomic selection. And by genomic selection, rate of inbreeding is also less as compared to other methods. In genomic selection, genetic gain is high because it increases the accuracy of selection, and it is the most reliable for the estimation of breeding values. And it also increases the selection intensity and decreases the generation interval because it applies early in the life of animal. And genomic selection is also very cost effective as compared to a traditional method like progeny testing. There are different traits that are used for the genomic selection. First, milk yield, fertility, mastitis, fat yield, protein yield, fat percentage, protein percentage, productive life, somatic cell count, also for the daughter pregnancy rate. But still there are some future prospects of genomic selection that there is a combination of more than one breed in a reference population. Means in a reference population, we can combine not only one breed, but more than one breed can be used as a reference population. And there is a national genetic evaluations to include genomic information because there is a genotype is also done in a reference populations. And also set a interbull organizations because as we know interbull which is situated in Sweden, that is used for the multi-trace across country evaluation MACE for the evaluation of breeding values of young sire or bull in a different countries. And by genomic selection, that is a setup for interbull organizations for comparing genomic proofs for different countries. Now, the approach to develop genomic selection methodology in India, there is basically three phases. First phase, learning phase. Second, building on learnings. And third, harvesting benefits by modified breeding designs. In first phase, learning phase, that is a development of custom chip for crossbred and indigenous cattle and development of buffalo chip. And in 2020, NDDB also produced a buff chip for the buffalo and also a high density chips also available in India. And in second, that is a building on learnings. In this phase, we can establish a genomic laboratory for the genomic study of an animal and whole genome sequencing can be done for cattle and buffalo breeds and develop a genomic estimated breeding value estimation of indigenous cattle breeds and buffaloes, and there is a routine estimation of breeding values. And third phase, there is a harvesting benefits by modified breeding designs, by modification of breeding designs and establishment of nucleus herd of allied animals. Now, finally, conclusion. As we know, genomic selection will surpass traditional selection as the dominant breeding program due to detection of specific haplotypes that is directly associated with production traits. For sustainable breeding goals requires the measurement and proper weighing of all relevant traits in selection index to enable simultaneous genetic progress in the desired direction of all traits of interest. And in terms of dairy cattle population, there is a wide genetic diversity in India. Thus, faster genetic progress is possible with high accuracy and shorter generation interval. Now, thank you, sir. If any question, please ask. Yes, now presentation is over. If any queries or question, please.
Okay, you leave the meeting. Okay. Now meeting is over. We will meet at 2:30 p.m. The next session.
तो अभी नहीं लगाइए, यहाँ ना लगाइए अभी।
If you're speaking, you're on mute.
Yes, sir.
Ma'am, are you speaking? Then you're on mute, right? Because you were on mute. So did you finish the?
No, sir. I have completed my presentation, sir.
So thank you, ma'am. I would want to take this moment to congratulate you on this session and thank you for this session.
Thank you, sir.
From NADCL and the participating institutions, thank you very much for this session. Thank
Thank
you,
you.
everyone. Thank you, everyone, for joining. Take care.